Hello friends, I am back with part 2 of plant cell culture and applications. Hopefully you have watched the video of part 1 of this particular chapter and have got benefited from it. Key areas covered in this part 2 video. Basic techniques of plant tissue culture, commonly used medium, types of cultures and plant regeneration pathways. Basic technique of plant tissue culture. First step is the selection of suitable explants like shoot tip, leaf, cotyledon, etc. Once the explant is chosen, surface sterilization of explants are done by using disinfectants like sodium hypochlorite, and later it is washed with sterile distilled water. Surface sterilized explants are transferred onto the suitable nutrient medium, which is also sterilized by autoclaving in the culture vessels under sterile conditions. Usually this transfer is done in laminar flow hood in the tissue culture laboratory. Growing the cultures in the growth chamber or plant tissue culture room. Fluorescent tubes are used to provide the artificial light of 16-hour photoperiod. Temperature is maintained as minus 26 degrees Celsius and relative humidity 50 to 60%. Regeneration of the shoots from cultured plant tissues and their elongation. The proper regeneration medium will be supplied towards this purpose. Rooting of regenerated shoots on rooting medium, which will have a lot of auxin because auxin promotes rooting. Transfer of the plants to the greenhouse or field conditions following acclimatization or tissue hardening of regenerated plants. So all these are the techniques of plant tissue culture. Coming to the commonly used nutrient medium. Most commonly used medium is MS medium or Murashige and Scoop medium. LS medium, B5 medium and niche medium are also used. Both solid and liquid medium are used and agar is used for solidification. Agar is obtained from red algae Gelidium amansi. Types of tissue culture. There are two types of tissue culture, organized culture and unorganized culture. In the organized culture, the culture of the whole or parts of the plant is done. The characteristics and organizational structure of a plant or organ is maintained. Examples are axillary bud culture, terminal bud culture, seed culture, embryo culture, ovary culture, and pollen culture. Coming to unorganized culture. There are different examples of unorganized cultures: callus culture, cell suspension culture, organogenesis, somatic embryogenesis, protoplast culture, etc. Let's see each one in detail. Coming to explant culture. Culture of the plant parts like stem, leaf, etc. It's generally used to induce callus or plant regeneration. The picture of the callus is given here. Callus is nothing but an unorganized mass of cells which are parenchymatous. It's very translucent. Auxins are added to the medium for callus induction. Callus cultures are used for number 1, plant regeneration, number 2, preparation of protoplast, and number 3, genetic transformation studies. Coming to cell suspension cultures. Single cells isolated from callus and are cultured in the liquid medium. Both mechanical and enzymatic methods can be used for isolation, even though enzymatic methods are preferred over mechanical methods. Mechanical methods can cause wear and tear. Cell suspension cultures are used for induction of somatic embryos, in vitro mutagenesis, production of secondary metabolites and genetic transformation. Next, let's look into mass cell culture. Large-scale culturing of cells in plant bioreactors which do not have stirrer is what we call as mass cell culture. We do not use stirrer because plant cells are very shear sensitive. Mass cell culture is important for producing secondary metabolites which are of pharmaceutical importance. Coming to protoplast culture. What are protoplasts? Protoplasts are cells without cell wall. They are isolated from plant tissues by enzymatic methods. It is cultured in suspension cultures in Petri plates, and they are used for various biochemical studies, producing somatic hybrids and cybrids and genetic transformation. Let's look at the difference between hybrid and cybrid. In case of hybrid, when you take two protoplasts of parents of different genotypes and fuse it, both the cytoplasm as well as the nucleus will be fused. But in case of cybrid, only one of the nucleus will be retained and cytoplasm of both the protoplasts will be maintained. That is why it is called cybrid. And the common enzyme which is used for the isolation of the protoplast is cellulase. Because cellulose is a major constituent of plant cell wall, so cellulase can be used for as an enzyme to isolate the protoplast. Coming to protoplast fusion. In this technique, protoplasts from different genotypes are fused with fusogen. Fusogen commonly used is polyethylene glycol PEG. Hybrid cells are selected by different antibiotic markers or fluorescent dyes which act as selection markers. Somatic hybrids thus obtained after selection are characterized by morphology and molecular studies like RAPD, random amplified polymorphic DNA. In this picture, we can see protoplast and protoplast fusion also. Protoplast fusion serves as a unique method to raise new genotypes with useful agronomic traits. Coming to plant regeneration system. There are two ways to regenerate plants. One is organogenesis and another one is somatic embryogenesis. Now coming to what is organogenesis. Shoots or roots are induced to differentiate from a cell or cell clusters. Usually, auxins promote rooting and cytokinins promote shooting. Coming to somatic embryogenesis. Totipotent cells undergo embryogenic pathways to form somatic embryos. New plants are formed from somatic embryos and they differ in their origin as well as large size from zygotic embryos. There are various stages of somatic embryogenesis which include globular, heart-shaped, torpedo and cotyledonary. Here we have a different stage of embryo development of somatic embryos. So you can see the globular shaped structure, heart shaped structure, torpedo structure and cotyledonary structure of somatic embryos. Here is a picture of organogenesis where we have rooting and shooting. For watching this video. If you like it, do click the like button and do subscribe to this channel for more upcoming videos on plant tissue culture and applications as well as other topics of biotechnology. Thank you once again.